to get cozy.
That's fine.
Just a quick word, Peter, and coach sharing is
Sorry, it's a little tight here.
Class is fine. Oliver and Jessica.
mobiles off the side, please. Can everybody please turn off their mobile phone on silent or off? Just quickly, Paul is going to make some very interesting remarks. The live webcast will start. The story is broken now it's running. Then she'll introduce everybody one by one, 3 to 5 minutes max. It's a tough chair, so if you go over 5 minutes, you'll cut me off. Okay? The idea is the journalists get a lot of time to run text. And then she'll open up to the floor, she'll make sure the journalists say where they're from, which organisation, and what else they're to. Okay? Just bear in mind that it is big webcast, so
So you representing Victoria for a minute.
Good morning, everyone. So welcome to you. I'm Paula Fujiwara. I'm the Scientific Director of the Union, your host for this conference. And I want to welcome you to the TB Science 2019. This is the press conference for this event, and that we hope will set the tone for the week ahead of all the exciting science that we're going to have. The Union is, of course, the custodian of the 50th Conference on Lung Health, taking place this week in Hyderabad. And I must say we're very delighted that the event is already making waves on global websites. I'd like to say a few things before I introduce my esteemed guests sitting with me here today. But before I do that, let me do some housekeeping. As Michael said, please turn your phones either off or to silent. So we are being webcast, so it'll be a little distracting. So we are going to be live streamed on the conference YouTube channel, and it's going to be archived, so you can also join available on the conference Facebook page later on today. And for those of you who are looking at us online, a very warm welcome to you. Today we'll be hearing brief remarks from all of our 7 speakers. You say we have a very crowded table today, and then I will open it up to your questions. We'll have microphones. You can see that there are microphones set up on both sides of the room. And if you'd like to ask a question, please line up behind the microphone. I ask that you state your name and the media outlet that you're representing and to whom you would like to address the question. Please keep your questions very brief. We have, as I said, we have very little time. And once the press conference is ended, you can speak one-on-one to any of the speakers that you wish to or arrange to do at a later time. Now, I do know that there's a conference going on downstairs, so we do realize that some of the speakers here do have to go back downstairs, so again, it may be a matter of arranging later. So let me say a couple things to briefly set the scene today. I think many of us were in New York in September of 2018 at the United Nations High-Level Meeting on Tuberculosis. And the political declaration that was signed there by the world leaders made it very, very clear that what we've all been championing here around this table for many, many years. To end the tuberculosis epidemic, we need a larger discussion this week around the role of prevention in the TB response. Prevention has for too long, I think all of us realize, has been too long neglected in the global response to the tuberculosis epidemic, and I'm delighted that this week it will take center stage in Hyderabad on the back of some various exciting research and announcements being presented here, not only at this meeting, but at the Union Conference later on this week. So we're also very delighted to see that this is 5 minutes, so we have time for questions at the end. And our first speaker needs no introduction. This is Mark Hatherill, who wears a number of hats and indeed has been involved in this M72 trial. But today we have him here as the TB Science 2019 International Co-Chair. He will provide a little bit of context and reflect briefly on the challenges of vaccine research and where the current state of investigation lies. So Mark, please go ahead.
Thank you. So I'm Mark Hatherill. I'm a professor at the University of Cape Town and director of SATVI, which is a research group based in South Africa that focuses on TB vaccine development. So as I reflect on these exciting results, and we're going to hear more details from Olivier and Anne, I'm conscious of the fact that we've been using the same tuberculosis vaccine, which is BCG vaccine, for almost 100 years. And the limitations of that vaccine strategy are clear when we look at the raging tuberculosis epidemic in developing countries. The highest bar for tuberculosis vaccines has always seemed to be protection of those individuals who are already infected. In other words, protecting those individuals from progression to disease. And that has been the main limitation of BCG, and it has been a goal to which we've been working through many years.
It's going to give us another perspective, the perspective of the person who has actually suffered from tuberculosis, and in this case has suffered from multi-drug resistant tuberculosis. He studied Diptendu Bhattacharya, has studied biotechnology in West Bengal and Chennai, and began working actually for a pharmaceutical company while also studying for his PhD. He had to stop his studies because when he underwent treatment for multi-drug resistant tuberculosis.
And above all, the cost of opportunity, which I lost, hurts me a lot. My family gone through a tremendous pressure during these days. My old father has to accompany me every day to the hospital for that thing. So I feel there is a compulsion from me to voice for the vaccine immediately right now, because it is a call of the time. There is a 4 cornerstone to stop an epidemic, I felt, like awareness, prevention, care, and cure. And this cure is not will not help to eliminate this whole epidemic once and on all. We must prevent it on the onset. Currently, the popular drug, which is my previous speaker Mark have mentioned, BCG, is in affordable space. But I myself also have been treated with BCG when I was a child. Still, I have got the disease. So it is important and call of time we develop a new vaccine for adults and young adults, so that this epidemic can stop for good. And I have gone through some papers, and I have studied that new promising vaccines are being created, and M72 is a promising one. I have heard of that. My next speakers will elaborate on that, I hope. And now few things I would like to add with this thing which come to my mind when we talk about vaccine. First, it should be affordable and cheap and available everywhere throughout where it is needed. Because when I was going through the treatment of MD at TB, I also had a lot of difficulties procuring medicine, because supply sometimes got detained and all those stuffs. So I had to check out for that much drugs in the open market, but it was not affordable, neither advisable, not affordable for every common people. Secondly, we must be patient. Mycobacterium goes very slowly, so we must be very patient like them, so that we can develop new drugs and will not stop the effort to develop better and better vaccines day by day. And we should push for the investment for that research, so that we can mitigate the opportunity lost and man-hour lost during the treatment period, which is very long according to my opinion. And also we have to do the trial properly, so that the protection of the trial groups is ensured by the government. Finally, I would like to thank Government of India, those they have called voice for Desh Jitega and TB Harega campaign. And but I would like to make my point very clear here, that we must not focus only on cure, but also the prevention. And then only this can be eradicated for good. Our group, Survivors Against TB, is still continuing to push for the agendas to the government, and I hope that case like mine will not be coming forth in the future very soon. Thank you.
Thank you so much for those really important and salient remarks. Our next speaker is Anne Ginsberg, and Anne is a senior technical advisor at the International AIDS Vaccine Initiative, or IAVI, who have of course, been partnering with GSK on the M72 vaccine candidate trial. She's going to focus on describing the design of this groundbreaking phase 2B study with the MS72 AS01E and talk to the top-line final efficacy results. Yeah.
Thank you, Paula. Good morning, everyone, and thank you for the opportunity to speak with you today. IAVI is very pleased to have partnered with GSK on this groundbreaking trial of a TB vaccine candidate that could potentially become a truly transformational tool in the fight against the global TB epidemic. This morning, we're announcing the final data from a phase 2B trial that evaluated GSK's vaccine candidate, M72 AS01E, for safety, immunogenicity, and efficacy in preventing HIV-negative adults who are already latently infected with the TB bacteria from developing active pulmonary TB, that is TB in the lungs, the place where it's most common for adults to get TB. The final analysis demonstrated that M72 AS01E had an overall vaccine efficacy of 50% during the 3 years after vaccination. These results are consistent with the primary analysis that was published approximately a year ago, and both the primary and the final results are now published in the New England Journal of Medicine. The trial itself was a randomized, double-blind, placebo-controlled study conducted at 11 sites in 3 TB endemic countries, South Africa, Kenya, and Zambia. It involved 3,573 participants, all HIV-negative adults between the ages of 18 and 50 years.
And then after that, we entered a long and very important collaboration with IRAS, which recently became the IAVI. And so along that road, we generated a lot of data in a lot of countries, including safety and immunogenicity data. But the most important data is the data that are presented today and published. And Anne was just explaining that we demonstrate that we can cut by 2 the number of disease in people who are already infected, which is a tremendous step forward to develop a new vaccine. So the vaccine seems to be very important. Is it safe, and how does it compare with what we have seen before in previous studies? Well, the safety profile of the vaccine is an acceptable safety profile. It means that we have a number of reactogenicity events that are in line with what we observe with other vaccines. Generalized 1 year ago, and now after 3 years of follow-up, we confirmed the previously observed results. Basically, with a plateau, the efficacy seems to be the same at year 3 than at year 2, and the antibodies as well seems to be at the same level. So, this is what I wanted to summarize today. Thank you.
Thank you very much, Olivier. Our next speaker, Matteo Zignol from Look Like. Thank you very much.
Thank you very much. Can everyone hear me? Thank you for this opportunity, and I'm sorry again that Matteo was not able, I don't want to echo, was not able to come and attend this very important presentation. The World Health Organization, again, is very excited about this phase 2B results of the M72 vaccine, and we've been working very closely with partners and collaborators and developers to see, to facilitate the next steps. One of the steps that we have taken, for instance, has been organizing a very high-level meeting, because WHO has a convening role among funders, governments, developers, to see what kind of both scientific and partnership and implementation partnerships can be taken going forward. In follow-up to this meeting, WHO is also now has organized a clinical trial design workshop to kind of get consensus between partners, developers, as well as countries. So when the next experiments and studies are being done on the M72 vaccine, the results get consensus and acceptability from all stakeholders. WHO will continue to support the development of the M72 vaccine studies, and once the results are confirmed of this very exciting findings, will of course then support member states adopt and implement the vaccine.
Thank you very much, Nabiat. And our final speaker is Peter Kim. He is the director of the Division of the AIDS Therapeutic Research Program at the National Institute of Allergy and Infectious Disease within the National Institutes of Health in the United States. And he's going to talk briefly to a new announcement by the NIH around TB vaccine funding.
Thank you very much, and thank you for the opportunity to come and speak here today. First, I'd like to just congratulate our colleagues at GSK, IAVI, and all the investigators that were involved in the M72 vaccine trial. I mean, the results are truly astonishing and results that are worth applauding and celebrating. We at the NIH do believe that an effective vaccine against TB is exactly the type of disruptive game changer that we need to bring the TB epidemic to an end once and for all. And towards this end, I'm here today to announce that we are going to fund 3 new research centers across the U.S. that are going to be committed towards answering the very question that was asked here today of better understanding the immunology or the immune response that is needed in all of us, and that needs to be elicited by a successful vaccine to prevent TB in all those that are at risk of getting TB. Basically, the first-year investment of these centers is going to be around $30 million, and we plan to actually continue that investment for up to 7 years to make sure that the research that we are funding is going to have an applicable impact towards the development of new vaccines for TB. These centers are going to help us better understand the immunology of TB vaccines, both in the laboratory setting, in animal research, and also in humans. So basically bringing all that research together under one roof so as to facilitate rapid advancement of research towards new vaccines that are going to help us bring this epidemic to an end. We believe this type of research will also provide the basis of understanding so that everyone involved in TB vaccine research, not just the investigators that we are funding, but everyone around the world will be able to use this knowledge then to apply towards their vaccine research to help advance their vaccine candidates faster through the whole development pipeline, first from discovery all the way through animal testing, then into clinical trials. We also think that this type of research is going to help us better inform the investments that we need to make in terms of vaccine research so that the investments that we are making are both efficient and effective. Lastly, I'd like to say that this type of research really depends on a whole wide range of collaborations, both among investigators and investors that are involved in TB research funding. And as you can see just from this group today, I think TB research is much more than just the individual scientists working in their lab to get something done. It incorporates actually a whole worldwide network of investigators, funders, community activists, and all those that are involved in TB research. And I'm here today to say that we really hope that this type of collaboration will be further engendered by these really promising results that we see from M72, and that this will really stimulate a global collaboration towards ending the epidemic and really towards research that are going to fund innovative tools that will help us bring the epidemic to an end. Thank you.
Well, that was a wonderful way to end the speakers, to say this announcement of new funding for vaccine research, which is sorely needed, because if we don't have new tools, we're not going to get to the end of tuberculosis, and vaccines, as we all mentioned, is really the key for prevention. So now, we're now open for questions. You will have microphones that are set up here and here, so I'd like you to go up to the microphone, please. And when I call, remember to state your name, state your media outlet, keep your questions brief, and indicate to whom you are addressing the question. So now open, please come to the mics. I do not believe that a group of journalists are not able or willing to ask questions. Here we go. Your name? Is the microphone on?
Can you hear me?
Yes,
Okay.
you can.
Hello, my name is Tabassum. I work with the Indian Express newspaper, a national daily from Mumbai, India. My question is directed to Mr. Oliver. You briefly spoke about the immune responses and how a TB vaccine works, and you said that there were high level of antibodies that were produced. Could you talk a little bit more about these 13 patients, the 13 people in the population who did get infected with active tuberculosis, and how does immune system factor in this? Because every person has a different immune system. So.
The way this clinical trial was designed is that only a subset of the subjects, a subset of the participants were sampled for immunogenicity assessment. The patients who develop tuberculosis, we don't have antibody levels for them at the moment. So in the current study, we are not able to make any kind of correlation between the antibodies and the actual protection. However, our partners at IAVI have been very wise to design, in parallel of our study, a biobank where basically almost all the patients in our study have given blood to that biobank, and the aim is that together with the clinical results, the biobank will be able to analyze in details how the immune system seems to actually generate this protection. And maybe Anne wants to say a word about the biobank.
Sure. So the biobank or biorepository collected specimens from 99% of the participants in the trial. So as Olivier said, that now provides the opportunity, and we're doing this in collaboration with the broader research field, to look at whether there are markers in the blood of people who are protected compared to those who were not protected, those 13 patients in the vaccine group and the 26 in the placebo group, to see if we can identify blood markers then that would correlate with being protected. So that's called a correlate of protection, and would be extraordinarily helpful in vaccine studies potentially moving forward, and make vaccine development much more streamlined, as well as, importantly, providing insights into what it is in the human immune response that is protective. So that's very exciting work that's just beginning to get off the ground now.
Yes. Microphone, can you turn up the microphone there? Yeah, it's on.
Is it on? Now
Yes.
it's on. My name is Anu. I'm a journalist in Delhi at The Wire. Just first question, is there a reason why this trial was not done in India, given that we have the largest burden? Is there something in Indian rules and regulations that make it difficult, or is there another reason? I'll just get through the rest.
Who's the question directed to?
Anybody, probably Mark or Olivier, yeah. And also, I haven't read the study in NEGM, but if I've understood this right, you're saying that a lot of the safety efficacy parameters have been in line with previous vaccines. If that is the case, what is the marked improvement of this over others? What is the marked improvement of this vaccine over the others? If so many of the parameters are just in line with the others, unless I misunderstood this completely. Yeah. And lastly, I just want to ask you, apart, because this entire press conference seems to be about this vaccine, are there any other companies, universities or institutes which are also doing very good work in the same area? Who are the other contenders or competitors in this field?
I will address the first two questions. First, when I said that the results were in line with previous studies conducted with M72 vaccine and not with other candidate vaccines, I was referring to the experience we at GSK and IAVI have developed over the past few years with M72 candidate vaccine. Why the study wasn't done in India? Well, this was obviously one of the options that we considered when we designed the study. The reason why finally we decided to go to sub-Saharan Africa was not related to regulations, as you suggested. It was related to mathematics. We needed a certain number of cases of tuberculosis to be accrued in the study. And the incidence rate, which is the number of cases in one geography divided by the population, the incidence rate in South Africa is about 10 times higher than the incidence rate in India, which means that if we had conducted the same study in India, the study would have been 10 times bigger, and so it would have been longer to achieve the results. That's the reason.
Does anyone want to comment on the issue of the other candidate vaccines?
Sure, I can do that. So there is a whole portfolio of vaccine candidates being worked on by a number of different organizations and partnerships for TB. There are currently about 15 different candidates at various stages of development throughout the world. This one is being talked about today because it's the first one to have these kinds of exciting efficacy results to show that a vaccine candidate can actually protect people, adults specifically, and those who are already infected with the TB bacteria, can prevent them from getting active TB. That's something that's never been shown before, certainly not in the published literature that I'm aware of. So that's why the excitement about this candidate today. But luckily, there are lots of other vaccine candidates coming along as well, because this is a very complicated epidemic. We need to be able to protect people all over the world, different age groups, HIV infected and uninfected, as well as those who are not yet infected, those like the ones in the study we did that are already infected, and even perhaps use vaccine candidates to help people who have active TB to shorten treatment or increase the cure rates from treatment, particularly for multi-drug resistant and extensively drug-resistant TB. So there are lots of different needs in this epidemic for vaccines. My personal view is it's unlikely that any one vaccine is going to address all aspects of the epidemic, and so it's important to have a robust portfolio of vaccine candidates moving forward.
Thank you, Anne. Yes.
Thank you. My name is Mary Mwoki from Standard Media Group in Kenya. In your trials in Kenya, did you take into account patients who have drug-resistant TB? And what was the efficacy of the drug in their case? Thank you.
Who are you asking the question to? To whom? Anne.
Sure. So this trial had to be people at baseline, right, who did not yet have active TB. And when someone is latently infected, as these participants were, one doesn't know for sure what strain of TB they were infected with, whether it's drug sensitive or drug resistant. So we expect that a vaccine like this, in all likelihood, will be effective in protecting against drug resistant as well as drug sensitive TB, but we don't know that yet. That's still a hypothesis. We are sequencing all the strains of TB that were identified from sputum from participants in this trial, so that in the end, when we have those data, we will at least understand whether the people who got TB are getting it from drug-sensitive or drug-resistant TB. But it'll be, that's a small number of participants. Those will still be small figures.
So we have time for one more question. Oh, yes, go ahead. Your name?
Jackie Thornton from the Lancet.
Yeah, I think you need to tilt the mic for you.
There.
Yeah, there you go.
Jackie Thornton from the Lancet. A question for Olivia and Anne. Obviously, you're very excited about these results. Can you spell out what are the next steps going forward? And you talked about other populations need to be tested, trialed. How close are we going to be seen to when this could be available, marketed?
Well, I'm going to ask Olivier to address that question because it's GSK's vaccine. So.
We have, in the room, we have our Chief Medical Officer, Thomas Breuer, who will address that question.
So first of all, obviously we are very excited as a company to have contributed to this very exciting results which were just published today. Your question is, what are the next steps? WHO has already alluded that there were two very important meetings at WHO which brought all the experts together, including all the countries which look for such a vaccine. India, South Africa, Kenya, everyone was there and essentially discussed what are the next steps, how to plan such a trial. GSK is fully committed to enable the future development of the vaccine. However, we want to do this in partnership. You might know that we have developed another vaccine against malaria, which is currently pilot tested in Africa. And based on this experience, we believe we have to define now true partnerships end to end. We have to decide how much vaccine is actually needed in the future if the vaccine is successful, where the vaccine should be manufactured. So a lot of questions based on our experience with the malaria vaccine which should be addressed now. So we are in active discussions with potential external partners to take this forward, and when we are ready to announce it, we will do that. Thank you.
Well, thank you very much. This is all we have time for right now. I just want to end by saying it takes a village, it takes a consortium, it takes the results of many to actually get to the end of what we're trying to do, which is to end tuberculosis. So thank you for joining us. As I said, some of the speakers have to go back down to the actual conference, but if you'd like, please come to the front and just arrange a time where you would like to speak with them. And again, thank you very much for attending the session.
Well done. Well done.